DNA replication. DNA replication, not black and white. Alright, since we've just learned about DNA and RNA and some of the differences between DNA and RNA, we learned a good amount about what makes up a DNA molecule, alright, the three parts that make up DNA and RNA. We're really going to be focusing on DNA right now because DNA is the storager of information that the cell and the body and the organism need in order to make all the things that they need in order to survive and do what they do. So we're really going to be focusing on deoxyribose nucleic acid in this first part. We will talk about RNA later on, but for now we're going to be talking about DNA replication. What we're trying to do is really get these, there's two strands of DNA here that are connected by hydrogen bonds. All right, and the hydrogen bonds are occurring between the nitrogenous bases of each strand. As you can see, one strand is going in one direction with the 3' hanging out always on one side, and it's 5' over here, and the other strand will go in the other direction. So this is the 3 to 5 strand on this, and this is the 3 to 5 strand of the other one. All right? But ultimately what we want to do is we want to replicate all this, and replication happens every time a cell wants to divide. So every time a cell wants to divide, it takes all of its DNA and it replicates the entire portion of DNA. So replication is a pretty important process that is also a fairly difficult process. But we're going to take you right through the process of DNA replication. First of all, it happens in the nucleus. All right? The nucleus is where the DNA resides. The DNA will never leave the nucleus. So that is where we want to replicate the DNA if the cell wants to divide. All right? Remember, replication of DNA is only happening during cell division. It's not happening anywhere else. Okay? So a cell looking to divide is going to have to replicate its DNA. So DNA here, this double-strand DNA connected by nitrogenous bases, hydrogen bonds in the middle. Here in the blue is the original strand. All right? The original strand is these two blue strands here. What's going to happen is each strand is going to act as a template for a new strand, so that the new DNA that we get, each one of these strands is going to attach to an original strand. So one cell will get this piece of DNA, and another cell will get this whole piece of DNA. So the resulting cells each will have a parent strand and a daughter, what we call a daughter strand, a new strand. And this is called semi-conservative replication. All right? It was once thought that it was completely conservative, meaning you'd have the old strand would completely replicate and then you'd have a completely red new strand of replication. So replication really begins when the cell receives a signal called growth hormone that signals something called TGF-beta, which then cascades and catalyzes reactions that are then going to express and unmethylate genes that are responsible for making these things called E2Fs, which are called transcription factors. And transcription factors allow the cell to go into replication because ultimately and normally the cell actually isn't replicating all the time. So as a result, the cell needs a positive mechanism to allow the cell to go into division when it's normally keeping the cell from dividing. Uncontrolled cells that do not control their cell division are called cancer cells. All right? So this stuff's really important. The transcription factors have a vital role in making sure that everything happens when it needs to happen. And we're going to talk a lot more about cancer cells in the future. But you should know that cancer cells don't have the same control mechanisms that normal cells do. The process of replication is enzyme driven. It has many different enzymes, has many different steps. It occurs in the nucleus. We just talked about it being semi-conservative. And in the end, DNA is completely replicated so that the cell can enter mitosis, most specifically prophase of mitosis, or meiosis for that matter. The enzymes that are used in replication are DNA polymerase 1, DNA polymerase 3, DNA ligase, helicase, topoisomerase, RNA polymerase, also known as primase, and single-stranded binding proteins, which are not enzymes at all, which most of these proteins are, except the single-stranded binding proteins are not. So these are essential for your understanding of the process of DNA replication. All right. Now, DNA replication starts when an enzyme called helicase, all right, is going to get inside the two strands, and it's going to rip apart the strands and separate the hydrogen bonds that connect the two strands together. Remember, one of the strands is going 3' to 5', and the other strand is going 5 to 3. That's really important here, as you'll notice. The end of one strand has a 5' end, which is a phosphate sticking out. Its 3' end is over here. And on this over here, if this is the 3' end, then this has to be the 5' end of the same strand. So replication, the parent strand is 3 to 5, but it's the strand that is made from it. All right, the daughter strand is going to be anti-parallel to it, because remember, we're resulting in two DNA double helices that are both anti-parallel to one another. All right, so it starts off with helicase, which creates the replication bubble, all right, which is this here, which is we have now ripped the strands apart from one another. All right, and the enzymes are going to start somewhere. It's called the origin of replication. And they're going to start moving and reading in the 5' direction on that strand. Now you remember 3 to 5 is important because these enzymes cannot read going in the 5 to 3 direction. They can only go towards the 5' direction of each sugar and the backbone. So if we start here, the replication origin in the replication bubble, the 5' end is to the right here on this bottom, so we have to go right. So first thing that's going to happen, helicase comes in. Second thing that's going to happen is an enzyme called RNA polymerase, or primase, is going to lay down a piece of RNA primer. All right, this green piece here. Okay, let's see if we have a picture, a better picture here. Actually, we're going to go to a video. Probably help you out a little bit. Nope, not the Michael Jackson video. That's another great video. Here you go, DNA replication.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from reannealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end.
In the 5'
DNA
direction.
Polymerase 3.
That's DNA polymerase 3.
No problem here, but the lagging strand presents problems.
What they actually didn't tell you, though, they didn't tell you that this strand, this continuous strand in the 5' direction, actually starts with an RNA primer, because DNA polymerase 3 cannot start from nothing. So it lays down a piece of RNA.
It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase 3.
Now remember, every time that RNA or DNA polymerase 3 wants to replicate DNA, it can only start from an RNA primer. All right, DNA polymerase 3 cannot start from nothing. So every time DNA polymerase starts, it has to start from an RNA primer, which is created by primase.
process repeats again and again. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragment.
So that should give you a little bit better idea of what's going on here. A couple of important things here. If this is the 5' end of this strand, all right, the top strand here, we're moving from right to left here because we always move in the 5' direction. So whatever started here at the origin, that red piece up here is called an RNA primer. An RNA polymerase has to lay down a piece of RNA so that DNA polymerase can go from that. All right? So RNA polymerase lays down a piece, DNA polymerase continues towards the 5' end. Now if you remember, if we get here on this strand, the 5' end is this way, onto the right. So we lay down a primer and we have one continuous strand. This strand here is called the leading strand on the bottom strand. And up here, this strand is called the leading strand. It's done continuously in one piece all the way to the end. All right? But remember, this is the 3' end of the top strand here. Our enzymes cannot move towards the 3. They can only move towards the 5. So over here, we have to lay down pieces of DNA replication. All right? And remember, DNA polymerase cannot start from nothing. So RNA polymerase or primase has to lay down a piece every time DNA polymerase needs to make a piece. Now, if you'd imagine, at the end you're going to end up with all these RNA pieces that you don't want, right? Because we're replicating DNA. So a new DNA polymerase comes in, DNA polymerase I, and it changes these red pieces, these RNA pieces, into DNA. Remember, the only thing it has to do is switch out a sugar, all right, a ribosugar for a deoxyribosugar, and change any U's, all right, to T's, thymine. So its job isn't really that complex, all right. So the leading strand is continuous in one direction, and the lagging strand is continuous in the other direction. All right, no, sorry. The leading strand is continuous in one direction. The lagging strand is replicating in the same direction, but it's jumping and going forward, jumping and going forward, jumping and going forward. Okay. Here are all the enzymes here too. The single-stranding binding proteins are all going to be attached to the strands to make sure that this replication bubble doesn't collapse and try to rebond with itself. All right. So here's a little some things about the lagging strand synthesis, which we just talked about. The lagging strand is going the 3 to 5, still going in the 3 to 5 direction, but we want to replicate this top strand here to the right. So we have to jump and come forward, jump and come left, jump right, come left, jump right, come left. All right. Whereas the leading strand, we can, if we're going towards the 5', which all enzymes can do, we can read and go in one piece and synthesize in the 5 to 3. Remember, everything's synthesized anti-parallel. Okay. Now, a lot of you might be freaking out right now. Maybe it's not something you remember easily from biology. We're going to talk about it in class, but it's also going to require a little bit of your help, a little bit of your questions. What specifically don't you understand? All right. If you need to, go back to the beginning and try to watch it again and see if maybe you can understand a little bit more. The lecture is over. You can stick around for an extra minute or two for a review of DNA replication for your help. So here are some things to remember. DNA replication happens in the nucleus. All right, that's number one. Number two, it requires a bunch of enzymes that are really important for its functioning. All right, all these enzymes are important. DNA polymerase I replaces RNA primers with DNA. DNA polymerase III does most of the replicating. Ligase fuses the Okazaki fragments, the RNA that was made into DNA, to the DNA strand. Helicase unwinds DNA to make the replication bubble at the replication origin. Topoisomerase is actually going to relieve stress on the rest of the strand. All right. If we have a replication bubble here, there's going to be more stress on these hydrogen bonds if we're ripping these apart. So topoisomerase will make sure that these strands don't come undone. RNA polymerase or primase is going to start replication. It starts by making, right, if we start here, this green piece is an RNA primer. The RNA primase is going to start from nothing. It's important because DNA polymerase cannot start from nothing. Okay? So it needs a primer. It always needs a primer. That's the difference between DNA polymerases and RNA polymerases. RNA polymerases can start from nothing. DNA polymerases need a primer. All right? Single-stranded binding proteins, again, make sure that the strands don't come back together actually after we created the replication origin. All right? So knowing the enzymes is important. Also, the third thing, knowing the difference between the leading strand and the lagging strand. The leading strand synthesizes towards the 5' end, all right? But it's making a 5' to 3' strand, all right? The lagging strand, on the other hand, still goes from 3' to 5' direction, but it's moving towards the 3' end generally. So it needs, if you're looking here, it's still synthesizing in the same direction, but it has to jump over in order to replicate all this stuff on the right of this top strand. All right? Because these strands are anti-parallel, the Okazaki fragments are occurring and happening on the opposite direction of this other strand. All right, if you look at that. All right. Knowing where they are, knowing what the enzymes are, knowing the general steps of replication and how it occurs are the most important things of this lecture. I look forward to your questions. I kind of jumped all over the place here, and for that I apologize. But for this homework assignment, and you're going to have a worksheet to do with it, just try to get the gist of everything and ask specific questions in class so that we can get this stuff ironed out. Thank you.